Hi guys, welcome to the second lecture in our series on the immune system. The aims of this lecture are to cover the adaptive immune system. We're going to go over the humoral and the cellular parts of this, so including different cell types and antibodies. We're then going to move on to the cause of other infections. Briefly, we're going to cover antibiotic chemotherapy with bacteria, and finally we're going to discuss a little bit about antibiotic resistance. This lecture is going to focus principally on adaptive immunity. This is the form of immunity which recognises and selectively eliminates foreign antigens. There are four key characteristics of the adaptive immune response, which are antigen specificity, a high level of diversity, displaying immunologic memory, which is the ability to respond to a second encounter of the same antigen more rapidly than the first encounter, and the ability to distinguish self from non-self recognition. There are major cell groups involved in adaptive immunity, being the B lymphocytes or B cells and the T lymphocytes or T cells. As a crossover between innate and adaptive immunity, we have the antigen presenting cells or APCs, for example dendritic cells. These cells express MHC class 1, present on all body cells, and MHC class 2, which is only ever expressed by antigen presenting cells and which allows them to interact with the T cells. So the first class we need to cover are the B cells or the B lymphocytes. These cells mature in the red bone marrow. They have a specific receptor called the B cell receptor, which is actually a membrane-bound antibody or immunoglobulin. A naive B cell, before any encounter of antigen, is shown at the bottom in the diagram, and you can see it has multiple BCR receptors or antigen binding receptors on its surface. At the first encounter of antigen, the B cells undergo a rapid division and differentiation into memory cells and effector or plasma cells. Memory cells have a very long lifespan and don't actually participate in this part of the immune response. However, the plasma cells produce a huge amount of antibody. This is essential for the specificity of the adaptive immune response. If you look at the diagram on the bottom, you can see examples of surface and soluble antibodies. You can see that they are identical, apart from the membrane component has been cleaved. You have heavy chains and light chains held together with disulfide bonds, and the antigen binding site is the top two parts of this Y-like structure. T cells, however, again are bone marrow derived but mature in the thymus. They express the T cell receptor on their membrane. They also express co-stimulatory molecules. Two essential molecules that they express are CD4 and CD8. T cells will only express one of these two molecules once they're mature. CD4 classifies a T helper or TH cell type, whereas the presence of CD8 classifies it as a T cytotoxic or TC cell. T lymphocytes recognise antigen that is bound to MHC. If a helper T cell is activated, this will lead to secretion of cytokines. The cytokines support the activation of B cells, cytotoxic T cells, and the activation of macrophages, hence why these cells are called helper T cells. To show you the difference, on the left we have a helper T cell showing the CD4 molecule in blue, the T cell receptor in purple, and MHC expressing antigen. T helper cells bind MHC class 2. However, cytotoxic T cells expressing CD8, shown here in pink, interact with MHC class 1 and antigen complexes. So it's not just the designation of the T cell that's important, it's because it affects which of the MHC antigen complexes that cell can actually interact with and recognise. brief bit of reminder for you on the antigen presenting cells. Remember, these include macrophages, B lymphocytes and dendritic cells, so are a cross bridge between innate and adaptive immunity. They express MHC class II. This allows them to interact with T helper cells, and they also produce cytokines which increase the activation of the T helper cells and so potentiate the immune response. Antigen presenting cells internalise antigen, break it down and then display a fraction of that antigen bound to MHC class II as an MHC class II antigenic fragment complex. When the immune system has been triggered and the adaptive immune system is triggered, we have two different classes of response. The first is the humoral response. In this type, we have B cells, which have differentiated into plasma cells and are now producing large amounts of antibody. The antibody binds to antigen, for example, as an effort of cross-linking, increasing opsonization and phagocytosis. The antibody may also activate complement, and if you need to remind yourself about this, have a look through the previous lecture. Antibodies are also capable of neutralizing bacterial toxins. It's usually the toxins produced by microbes that make us sick rather than the microbe themselves, so this is an essential function of the humoral response. The second branch of the adaptive immune system is the cellular response, and this is T cell mediated. The helper T cells, once activated, secrete cytokines. This increases the activation of other cells, including B lymphocytes and phagocytes. Cytotoxic T cells are now capable of eliminating altered self cells, for example virally infected cells and tumour cells, which are now expressing on their MHC class I various proteins which are no longer recognisable as safe and self. In the humoral response, the key effector here are the antibodies. Antibodies are immunoglobulins, and we find the gamma globulin fraction of the serum contains these compounds. Antibodies consist of 2 light or L chains and 2 H heavy chains, 22 kilodaltons and 55 kilodaltons in size respectively. They form heavy and light chain heterodimers with disulfide bonds. And we actually have a situation here called the dimer of dimers, as we have 2 heavy and light chain dimers bound together. Immunoglobulins have a variable or V region of around about 110 amino acids. This is the variable region, however the most difference is actually found in the CDR, which is the complementary determining region. This is the antigen binding site. The constant region has a constant sequence and is involved in the structure of the antibody rather than recognition of antigen. There are 5 major antibody classes determined by the difference in the heavy H chain. The constant region variation changes the isotope from mu, delta, gamma, epsilon and alpha. The heavy chain, however, determines the class of the antibody. So IgM, for example, is isotype mu, IgG is gamma, IgA alpha, IgG delta, and IgE for epsilon. And we'll now have to work through all 5 of these classes individually. Before we start on that, this shows you a structure of a classic antibody with the characteristic Y shape. You have the light chains and the heavy chains. The 2 heavy chains are shown with a variable and constant region, and you have heavy chains shown with the H regions. You have a variety of disulfide bonds formed between the 2. The key thing to remember here is that the antibody binding sites are at the end. The effector activity is actually determined by the constant region rather than anything else. Heavy chains are either mu, gamma, alpha, delta or epsilon, the light chains are always kappa or lambda. We can digest antibodies using enzymes to allow us to investigate their properties more thoroughly. We produce Fab fragments and FC fragments when undergoes pepsin digestion. So first of all, the Fab fragments are the antigen binding sites. So if you digest this structure with pepsin, it will give you Fab fragments and an FC fragment. The FC fragment binds to the B cell and contains the H chain only. We can actually use mercaptoethanol to reduce the disulfide bonds to produce two fragments here. This is very useful if we want to do research on the antibody structure, but you don't need to worry about this in terms of function. Over the next few slides, we're going to go over each of the different immunoglobulin classes in turn, starting with immunoglobulin G, IgG. IgG comprises over 80% of your total immunoglobulin complement and is split into 4 subclasses. If you look at the IgG structure, you can see how you have the hinge region and the disulfide bonds. IgG 1, 3 and 4 have the hinge region and then a large number of alternating disulfide bonds down the length. IgG3 has a far longer set of disulfide bonds than the others, whereas IgG2 has disulfide bonds above the cross as well as below. IgG1, 3 and 4 are all capable of crossing the placenta and are important in giving early life immunity to a newborn before that newborn has had a chance to develop its own antibody repertoire. Of these subclasses, IgG3 is the most potent complement activator of the 4, and IgG1 and 3 both capable of binding to the Fc receptors on phagocytes. This allows them to partake in opsonization, as the IgG has effectively coated the bacteria, bound them together, and is then able to bind the phagocyte to allow opsonization to occur and destruction of that complex. IgM is the next most frequently encountered antibody. It's 5 to 10% of the total complement and is found either membrane bound on B cells or as a pentamer of 5 IgM molecules held together with disulfide bonds. IgM is the first antibody of the primary response, has a very high valency and is extremely effective in agglutination and a neutralization of viral infection together with complement activation. The relevance of it being the first antibody to be found at the primary response we'll come on to later. IgA comprises 10 to 15% of your total immunoglobulin content and is found predominantly in secretions such as milk, saliva, tears and mucus. It's very important for immunity in the gastrointestinal, respiratory and genitourinary tracts. It forms a dimer with a J protein chain, and this allows it to have a secretory component. IgA prevents the attachment of bacteria or viruses to the mucous membranes, and again, along with the other antibodies that are capable of moving across the placenta or are present in secretions, is very important in protection of newborn infants. IgA can have a secretory quality to it. If you look in this particular diagram, you have the submucosa and you have a plasma cell secreting antibodies, in this case dimeric IgA. This binds to a polyimmunoglobulin receptor before being internalised in the epithelial cells, cleaved via enzymes and removed as secretory IgA into the lumen, together with that J chain allowing those two parts to form. The final two immunoglobulin classes are IgE and IgD. IgE has a very low serum concentration, however is essential in mediating immediate hypersensitivity reactions, allergies. It binds to the Fc receptors on the basophils and the mast cells, If you remember from the last lecture, the basophils are very granular cells. This binding of allergen causes degranulation to occur, and that's what mediates this immune response. So if you suffer from hay fever, for example, because your IgE is binding to the basophils and mast cells, once pollen binds, you get degranulation, histamine release, and you have the classic symptoms of an allergic response. IgD is very low concentration at 0.2% of the entire immunoglobulin complement, which corresponds to around about 30 micrograms per ml. The major membrane-bound immunoglobulin is expressed by mature B cells. We haven't yet identified what its function could be. This diagram shows you more about what I've just talked about with IgE-induced granulation. So here's a mast cell, the granules once again in the cytoplasm. The allergen, in this case pollen for example, binds to the IgE, which is bound to the Fc receptor specific for IgE on that mast cell. This causes degranulation and release of granule contents. Histamine is one of the most commonly known substances that mediates allergic reactions. It isn't the only one. However, it is responsible for a lot of the inflammation, runny eyes, watery eyes, and sneezing that we get with a lot of allergies. Antibodies have several effector functions mediated by antibodies and antigens. Once the antigen recognition by the antibody, the immunoglobulin, occurs, that invokes the immune response. The ultimate aim is to remove the antigen or eliminate the pathogen. Antibodies can do this in several ways. First is opsonization inducing phagocytosis, complement activation by C3b fragment binding, and this antibody-dependent cell-mediated cytotoxicity or ADCC. This is where we have binding of our antibody and immune globulin to NK cells, and it is specific to NK cells. Transcytosis can occur here, as well as transfer of immunoglobulin across the epithelial layer, for example IgG and IgA being transferred to the fetus. As I mentioned before, this is an essential way that newborn infants are protected by the mother's antibodies. Within our bodies, the diversity of antibodies that we can produce is immense. Both the heavy and light chains contain a variable region at the N-terminal and constant regions at the C-terminal. This means that we can have different isotypes, different classes of immunoglobulin, with the same antigen specificity, because a variable region can associate with a different heavy chain constant region. So we can have an antibody specific for a particular antigen of class IgM and IgG, depending on which associated region is associating with which constant region. There were several theories covering antibody diversity. The first was the germline theory, suggesting that germ cells contain a large number of immunoglobulin genes. The second was the somatic variation theory, strongly suggesting that our genome contains only a small number of immunoglobulin genes, and that the antibody specificity arose through mutation or recombination of these genes. In general, the one gene equals one polypeptide theory is accepted across the scientific community. In 1965, Dreyer and Bennett discovered two genes that encode an H or L chain. One codes the V region, while the other codes the C region. Then in 1976, Tonegawa and Hazumi discovered that during B-cell differentiation, the variable and constant genes underwent a rearrangement. This seemed to provide evidence for the somatic variation theory, and actually won Tonegawa the Nobel Prize for Science in 1987. When you look at the antibody coding genes, we have three here. So the first is the lambda chain DNA, second is the kappa chain DNA, and third is the heavy chain DNA. So you have variable regions shown as V, joining as J, and constant regions as C. In the kappa chain, you can see you have a totally different structure for where these various regions are located, and the same again for the heavy chain. When gene rearrangement occurs, for example, here's a germline kappa chain DNA, the variable region in the light chain is coded by two genes, V and J. Those different V and J genes can be fused. So if you have a look to the second part of the diagram, here we've rearranged that kappa chain DNA. The primary RNA transcript is then processed, and you have polyadenylation and RNA splicing, together with a poly-A tail added to give you an mRNA. Once translation occurs, you have a nascent polypeptide and a final kappa light chain, consisting of a particular V, J and C segment. The variable region in the heavy chain is coded by 3 genes, rather than the 2 coded in the light chain, and here we have a variable diversity and joining sections here. You don't need to worry any more than that about how somatic recombination occurs. It's something that we can go on to in stages 3 and 4. When we look at the immune response, we start to realise how important it is to our survival. This diagram shows viral, bacterial, parasitic and fungal infections and the infectious diseases with the annual deaths. And you can see a total number of deaths in any one year. This was around about 2006-2007. About 14.9 million deaths were due to infectious disease. Compare that to 7.1 million due to neoplastic, i.e. cancer, 5.2 million due to injury, 3 million due to asthma and chronic obstructive pulmonary diseases, 16.7 million were due to cardiovascular conditions, with all other causes of death, however weird and varied, taking up the rest of that pie chart. When you look at it, that means that almost the same very similar numbers of people died from cardiovascular conditions as died from infectious disease. If you look at the table, discover that actually 1.12 million of those deaths were due to vaccine-preventable childhood diseases. There's been significant problems in vaccination procedures over the last few years, ever since a link was suggested between the MMR vaccination and autism. This has actually resulted in a significant drop in vaccination take-up in school-aged children. A lot of parents rely on something called herd immunity, which is where the majority of children are immune either through natural reasons or via vaccination, and due to that, children who are not immune are effectively protected. Sadly, that is not the case, and the last few years we've seen a dramatic rise in cases of whooping cough due to the Trussis bacteria. We've seen a large increase in measles occurring. When we look at the different type of infectious diseases, the most problematic are respiratory infections, accounting for almost 4 million of those annual deaths. Briefly, before we go on to do viruses in the next lecture, I need to just give you a little bit of a heads up. So viral infections. Viruses are simply small nucleic acids with proteins and a lipoprotein coating. They are not, strictly speaking, alive. They require a host cell in order to replicate. They can coexist with that host cell or they can kill the host cell. The virus needs to be able to replicate and spread. For example, facile transmission, so think influenza virus. All know if you have a cold or a virus and you sneeze, you will spread that virus. Viruses tend to like mucosal membranes, so passage of the mucosa in the GI tract, respiratory tract and genitourinary tract are all at risk of viral infection. We know antibiotics can't treat viruses. However, our body's immunity is quite effective at actually clearing viral load. First response being the humoral response, affected by antibodies, especially secretory immunoglobulin A. IgG and IgM are also involved here, and actually complement activation by IgG or IgM are very effective at clearing these virally infected cells. The activities of these immunoglobulins include blocking binding of the virus to the host cell, therefore preventing that cell from gaining entry to the host cell and causing infection. It can block fusion of the viral envelope with the host cell's plasma membrane, enhance phagocytosis of viral particles, viral agglutination, and opsonization or lysis of an enveloped viral particle via the membrane attack complex of the complement cascade. On the other side of the adaptive immune response, cell-mediated, interferon gamma secreted by helper and cytotoxic T cells has direct antiviral activity and will destroy viruses. The cytotoxic T lymphocytes will kill virally infected self-cells because they recognise that the self-proteins have been taken over by that virus. Finally, NK cells and the macrophages will kill virally infected cells via antibody-dependent cell-mediated cytotoxicity. Remember that this is specific to the NK cells. To give you an idea of how interferons have an antiviral activity, here you have the interferon alpha-beta receptor bound to interferon. There are a variety of cascades, and you don't need to know the full cascades. You do need to know that there's a significant need for ATP in this process. Ultimately, you end up with inhibition of protein synthesis, and we now actually give interferons as a way of attacking viruses in patients, for example with swine flu. Bacterial infections are rather different. Immunity is achieved via antibodies, except for intracellular bacteria. The antibodies, together with complement, can cause opsonization. So if you look at the diagram on the right, you can see bacteria invading, damaging the skin, producing toxins. Complement activation precedes antibodies are actually starting to be produced. The immunoglobulins are now binding the toxin and neutralizing the toxin, as well as starting to opsonize the bacteria. Complement, in addition to opsonization and phagocytosis, mediates lysis via the membrane attack complex. Intracellular bacteria are dealt with slightly differently, as the innate immunity, so NK cell activation, and cell-mediated immunity via the T helper cells, are involved in destruction of the whole cell. Before we leave bacterial and viral infection, I want to just go over this table, which has been taken from Kubi's Immunology 6th edition. And this shows how we have various mechanisms for bacteria to infect our cells, how we can protect ourselves from that, and how bacteria have evolved to evade those mechanisms. So the first mechanism by which infection can occur is attachment to the host cells. Our host cells can actually block this via secretory IgA antibodies. However, a large number of bacteria produce proteases which cleave secretory IgA, and there is a significant antigenic variation in the attachment structures of the bacteria, meaning that we can't block all of them from attaching. Proliferation is a second major infection process, which can be prevented or protected against via phagocytosis, complement-mediated lysis, and the localized inflammatory response. However, bacteria have managed to evade these mechanisms via producing surface structures such as a polysaccharide capsule that either inhibit phagocytic cells, or they've developed mechanisms to survive within the phagocytic cell. Some species of bacteria, such as Shigella, will actually induce apoptosis in macrophages if they have been taken in to that macrophage. And there's a generalised resistance of gram-positive bacteria to complement-mediated lysis, which causes a significant problem. Insertion of the membrane attack complex is, generally speaking, a good way of destroying bacteria. However, it can actually be prevented from inserting into the membrane by the long side chains on cell wall LPS present on some gram-negative bacteria, for example E. coli. Host tissue invasion is a major infection process by which the bacteria actually get access to the tissues. Antibody-mediated agglutination here prevents those bacteria from accessing the tissues themselves. However, again, a large number of bacteria, the Pseudomonas class, secrete elastase, which inactivates the complement components C3a and C5a, reducing inflammation and reducing agglutination. Finally, toxin-induced damage to the host cells can be prevented via neutralization of toxin by antibody. But again, secretion of hyaluronidase via bacteria can enhance the bacterial invasiveness, and although it doesn't stop the neutralization of toxin by the antibody, it can rapidly overwhelm the host's defences. Of the last two pathogenic classes, the first we want to deal with are the parasitic diseases. Parasites can form either from protozoans, which are unicellular eukaryotes which must reside and multiply within host cells, and the helminthic parasites, which are the unicellular organisms such as gut worms, which can infect humans but are fully able to reproduce and survive on their own. The vast majority produce a chronic disease with the exception of an acute malarial attack. Malaria is caused by the parasite Plasmodium falciparum and has a significant health issue worldwide, with 10% of the population infected, responsible for 1 to 2 million deaths per year. The female Anopheles mosquito is actually a crucial part of the parasite's life cycle, and once biting humans will inject the parasite into the blood, where we form sporozoites, merozoites and gametocytes in the humans. We can mount an extremely poor immune response to the malarial parasite in humans, requiring significant amount of preventative medicine if you're heading to an area where malaria is present in order to try and prevent or at least reduce the risk of infection. In the mosquito's gut, the very first stages of the parasite's life cycle occur. Once injecting into the patient, in the human, they form sporozoites. These then migrate to the liver, where they form merozoites as the next stage of the life cycle and form capsules within the liver. The merozoites then rupture their vesicles and go out into the blood cells, where they infect red blood cells, multiply, and eventually rupture the red blood cell. This is one of the reasons for causing the problems. Meanwhile, while this is going on, some of those cells from the red blood cells become gametocytes, the male and female gametocytes, which are then taken back up when a mosquito bites that patient, and the cycle starts again with the gametocytes starting off the life cycle once again in the mosquito's gut. The final pathogenic class are the fungi. There are over 1 million species of fungi, of which only 400 are known to cause disease. We use fungi in practice, for example for fermentation to produce things like cheese, wine and bread, also for the production of penicillin. Our innate immune system is actually very good at controlling fungi, for example via phagocytosis by neutrophils, activation of the lectin and alternative pathways of the complement cascade, and via toll-like receptors 2 and 4. Fungal disease can be classed based on virulence, route of acquisition and site of infection. For site of infection, you can have superficial and cutaneous, which usually involve the epidermis, or the hair, nails and skin. Subcutaneous fungal infection tends to involve significant inflammation and tends to be associated with wounds, whereas deep and systemic infection involves the lungs, abdominal viscera, bones and potentially central nervous system, and forms far more of a problem for the patient. Fungal infection and fungal disease can be acquisition via exogenous means, so through the environment or from the skin, or endogenous, where the fungal infection has come from within the host themselves. Usually this can be latent reactivation or a commensal organism, for example on the skin, where the balance of commensals has been shifted. Virulence is either classed as primary or opportunistic. Primary virulents are inherently virulent and will infect a healthy host, whereas opportunistic fungi are low virulence and will only affect an immunocompromised host. That means patients who are already sick or immunocompromised in any way are going to be at far higher risk of infection via fungi than the general healthy population. In addition to these conditions that we know are caused by these pathogens, we're starting to see a problem with re-emerging diseases that were once considered to have been eradicated. Conditions such as TB or diphtheria, for example, that are very uncommon in the UK, are starting to return. We're seeing fatal infectious diseases such as Ebola in Western Africa and the West Nile virus, but we're also starting to see these conditions spreading to areas they weren't originally in before. In 2002, there was an outbreak of a condition termed severe acute respiratory syndrome, or the SARS virus. This happened in Asia, in areas of very high population density, and was a major health concern for quite a long time for the World Health Organization. In addition to SARS, we're seeing bird flu, the H5N1 influenza strain. As long as this is confined to birds, humans should be safe. However, there is a potential pandemic if combined with a human influenza virus, which would allow rapid cross-species transfer. The human immune system is incredibly effective at dealing with infections. However, in the face of such aggressive infections as TB, for example, Ebola, you need to have further assistance for the immune system. Although antibiotics are no good against viruses, they are very effective as chemotherapy against bacterial infection. And the remainder of this lecture is going to move on to discuss some methods by which we can choose a selective chemotherapy and by which bacteria can actually become resistant to that chemotherapy. When it comes to antibacterial chemotherapy, both infection and therapy are dose related. So we've got to think about the dose, the selectivity of the therapy we use, the therapeutic index, and whether we want to look at a bactericidal or bacteriostatic medication. We also need to take into account minimum inhibitory concentration, so the minimum dose of a drug which will inhibit that bacterium, and minimal bactericidal concentration, so the lowest dose at which bacteria will start to be destroyed. Different antibiotics have a different spectrum of activity, and many of them come with some contraindications, so cannot be used by patients on other drugs, for example. As a very brief overview of bacterial cell metabolism, you have effectively three different classes of reactions. The first are the class 1 reactions, where very, very simple molecules such as glucose and phosphate are combined together with other very small molecules to form precursor molecules and ATP. Class 2 reactions then take these molecules to create larger structures such as nucleotides, amino acids and hexosamines. And finally the class 3 reactions, which are going to take these substances to create peptidoglycan, proteins and the RNA and DNA that bacteria requires. As a result, the best targets for chemotherapy are going to be the class 3 reactions. These are where the small molecules are being assembled into much larger biomolecules. They're good targets because every cell must make its own biomolecules, and there are very distinct differences between prokaryotic and mammalian pathways. Both class 1 and 2 reactions occur in prokaryotes and mammalian cells, so you're at risk of causing far more harm than benefit if we target those. This slide gives you a summary of targets and antibiotics. So first of all, at the top you can have inhibition of cell wall synthesis, for example, via the penicillins, cephalosporin. You can inhibit protein synthesis with drugs such as chloramphenicol or erythromycin, tetracyclines or streptomycin. Cells need enzymatic activity for the synthesis of essential metabolites, so we can inhibit this process as well. For example, sulfanilamide or trimethoprim are effective here. Alternatively, we can injure the plasma membrane with a drug called polymyxin B, or we can inhibit nucleic acid replication and transcription, for example with the quinolones and rifampin. You don't need to worry too much about this, as this is providing you with the background material you'll need for stage 2. Bacteria can have intrinsic and acquired resistance. So intrinsic resistance are inherent features which prevent antibiotic action. They're usually determined and expressed by the genetic material contained within the chromosome. For example, beta-lactamases in gram-negative bacteria are capable of breaking down and inactivating the beta-lactam class of antibiotics, which includes penicillins. Acquired resistance is where resistant strains emerge from a previously sensitive population of bacteria, and this is usually determined by the acquisition of new material in the form of plasmids or structures called transposons, or mutations in the chromosomal genes. It is possible for bacteria to have both intrinsic and acquired resistance within the same cell. One way that bacteria can pass on an antimicrobial resistance gene is via horizontal gene transfer, and you have 3 methods by which this can occur. The first is bacterial transformation, where a donor cell releases its DNA, it can be lysed, releases the DNA together with the antibiotic resistance gene, which is then taken up by a recipient cell and incorporated into their genome. Bacterial transduction is where we infect a cell with something called a phage particle. That phage is then released by the cell. It's already containing that antibiotic resistance gene. It then injects its nuclear material, its genetic material, into the recipient cell, and once again that gene then becomes incorporated into the new genome. Bacterial conjugation is where a plasmid or a transposon can transfer from a donor cell to the recipient cell, where the 2 cells directly join together and send through that genetic material. I mentioned these structures called transposons. They are literally termed transposable elements, and they're discrete genetic elements capable of moving within the bacterial genome or from one DNA molecule to another. They are not capable of independent replication, so must be incorporated within the genome somewhere. This transposition provides multiple copies and is a very effective way of passing antibiotic resistance information and genes from one cell to another. So why should we be concerned about this rise in antibiotic resistance? One of the reasons that we should be very concerned is that we have a limited pipeline available for antibiotics. When you consider that we are now seeing resistant strains of Staphylococcus aureus, so MRSA, C. difficile, Enterococci and TB, we're reaching a point where we cannot provide sufficient antibiotics to treat these infections effectively. One of the reasons for that is that large pharmaceutical companies are less inclined to put in lots of money, several millions of dollars or pounds, to develop new antibiotics when they know that within 5 to 10 years that antibiotic is going to be redundant. We're seeing changes in population and social behaviour, so sexually transmitted diseases are being seen far more frequently in clinics than they have been in the past. And again, we're seeing a lot of problems with drug resistance in these conditions. To give you some examples of why we're so concerned about this rise in resistance to antibiotics, when we look at the bacteria Staphylococcus aureus, over 90% of the strains produce the beta-lactamase enzymes. This gene is plasmid encoded, conferring resistance to penicillin, ampicillin and the other beta-lactams. However, the strains were mostly susceptible to penicillinase-resistant penicillins such as methicillin, as well as some other drugs still, like first-generation cephalosporins, beta-lactam, beta-lactamase inhibitor combinations. However, in the UK, over 40% of Staph aureus bacteremias that were reported in the UK are now resistant to methicillin, as well as the other beta-lactams. And this gives us MRSA, or methicillin-resistant Staph aureus. The MRSA chromosome contains a 50 kilobase region called the MEK region, which isn't present in methicillin-susceptible Staph aureus. Unfortunately, most MRSA bacteremias that we see now are multiply drug resistant, and this is termed MDR MRSA, but are still susceptible to vancomycin. Vancomycin was traditionally thought of as the antibiotic of last resort. It was the antibiotic that you only bring out when everything else has failed. The reason for that is that we need to preserve susceptibility to vancomycin for as long as we possibly can. It is inevitable, almost, that resistance to vancomycin will occur and will come across universally. TB is a major health problem worldwide. 2 billion people are affected, and the world population is around 6 billion. That gives you an idea of how many people are actually affected here. There are 9.2 million new cases and 1.7 million deaths 10 years ago in 2006 due to TB, and in that year it was responsible for over 25% of preventable adult deaths. In 1993, the World Health Organization declared TB to be a global emergency, and that hasn't actually changed very much. They estimated that in the 18 years between 2002 to 2020, there will be 1,000 million infections, 150 million people will become ill, and 36 million people will die as a direct result of TB. In this country, we believed that TB had been eradicated by a careful health screening and vaccination programmes. However, in 2003, our UK TB cases increased 10% over the last 10 years, which started to give clinicians significant cause for concern. One of the reasons for TB being declared a global emergency is that we are more increasingly seeing multiple drug-resistant TB coming through. We know it causes 26% of all preventable adult deaths globally. However, 13% of new cases are resistant, with around about 1% resistant to both isoniazid and rifampicin. This is a major problem, especially for patients who are already immunocompromised, as they simply cannot shift the infection. We've now discovered that the TB bacterium has resistance mechanisms for all available therapies, has been elucidated. This means that although one strain may not be susceptible to drugs X and Y, but susceptible to drug Z, another TB bacterium may be susceptible to drug X, but not Y and Z. So this is causing us major problems, and typically resistance is a direct result of non-compliance. Therapy for tuberculosis is very long, a couple of years, with lots of different drugs and very complicated drug regimes. In underdeveloped countries or places where people do not understand the importance of drug compliance, patients start to feel better, so stop taking their drugs. That unfortunately means that bacteria that are susceptible can acquire resistance genes, and we're now seeing strains of TB classed as extensively drug resistance, or XDR TB, and they are a genuine emergency. To really ram it home how serious this is, we're seeing multiple drug-resistant TB appearing, or rather re-emerging, across Europe, Africa, Asia and the Americas. Just by looking at this chart, this is a very old chart now, so it is out of date. However, it gives you an idea of what's happening. So we're seeing vancomycin-resistant Staph aureus appearing in America, for example. Drug-resistant malaria coming in Africa and Asia. There are newly emerging diseases as well, such as the SARS virus, known in Asia, and we have E. coli strains and new influenza strains popping up. You have to remember from that first lecture on innate immunity and pathogens that bacteria and prokaryotes have been around far longer than we have, so it makes sense they're going to have good resistance mechanisms already in place for many of the drugs that we can produce. However, drug compliance is a major factor which is causing a lot of this resistance problems from coming, and you'll do far more of this in stage 2. As always, if you need any further help, please don't hesitate to contact me.